Good morning from Toronto, Canada. Hi, I'm Dr. Greg German, Chair of Global Clinical Phage Rounds. We have a most exciting presentation today, and it is a community forum event. I'm very excited to have Dr. Jean-Paul Pirnay and team talking about their magnificent article. I'll be giving a quick highlight to those who are familiar with Global Clinical Phage Rounds as a refresher, and those who are not as a opportunity. Global Clinical Phage Rounds started approximately 2 years ago. We're now in our 23rd session, and our sessions include those who are talking about grand rounds-like format for difficult phage therapy cases. And that's what 21 of our cases have been about from across the world. And as new countries get on board, they not only talk about their cases, but how they're trying to bring phage therapy forward. The second type is more of an industry engagement or a clinical trial engagement process, and those are hyper confidential. Where Global Clinical Phage Rounds, we have a network where we can have video, pictures, and other discussions take place after the event because we can't be around the globe all at the same time. Otherwise, you're up at 3 a.m. in the morning. And then these hyper rounds, which are at the request of industry, are very exclusive, not recorded, and felt to be hyper confidential. But just the opposite of that is where we want to have a community engagement event, and that's where we are today. I want to say that this is going to be, I think, a fantastic session. We have a great deal of participants that are already coming in. There's 4 in the waiting room. We have to admit them all. Okay, great. So let's go ahead and start with the introductions for today. So Jean-Paul Pirnay is an industrial engineer in biotechnology. In 1993, he joined the Queen Astrid Military Hospital, where he contributed to the development of human cell and tissue banks, including tissue engineering and cell therapy projects. In 2003, he obtained a PhD in medical sciences from Brussels Free University in Belgium, and in 2005, he was appointed as director of the newly established Laboratory for Molecular and Cellular Technology of the Queen Astrid, a position he still holds today. Lab MCT harbors human cells and tissue banks, a phage research laboratory, and a clean room production facility for human cells and tissue products and therapeutic phage preparations. He is, as I understand, also the officer for the ESGNTA, which is the phage therapy working group for the European Microbiology Infectious Disease Network. In addition, we are joined by Dr. Sabrina Green. She's a microbiologist with a background in phage therapy research, antimicrobial resistance. She earned her PhD in microbiology from the Baylor College of Medicine in Houston, Texas. During her graduate training, she co-founded Taylor, which stands for Tailored Antibacterials and Innovative Laboratories for Phage Research. She also helped us in our first case in Canada for urinary tract infection, for which we're always grateful. And this is Taylor is a nonprofit service center at Baylor dedicated to sourcing and repairing phages for patients with difficult to treat infections. Following her PhD, she served as Taylor's Director of R&D. Dr. Green is currently based in Belgium, where she works as a postdoctoral research associate at the Lab of Gene Technology. Her recent research focuses on bacterial genotypic and phenotypic changes associated with phage resistance following phage therapy in collaboration with the Queen Astrid Military Hospital. And then last but not least, we have Sarah Djebara, a medical doctor specializing in internal medicine, mainly on systemic and autoimmune disease and on tropical medicine. She is part of a University Hospital St. Pierre in Brussels, and part of her time is spent at the Queen Astrid as a medical doctor responsible for the phage therapy since 2007. So I'm going to stop sharing my screen and at the same time offer that we have 2 excellent co-discussants today, Dr. Ranier Paz from the ESGNTA chair, and Dr. Gina Sa from the Mayo Clinic, vice chair of Global Clinical Phage Rounds, and as well, they're both fantastic people. So as we move forward, we're going to admit more people to this, and do know that these are going to be public events, opportunities, meaning that this is recorded and potentially shared with others. So keep that in mind as you share your comments today. At the end of this, we'd like to have everyone take a big, huge picture, if you will, so we can share in the joy of Global Clinical Phage Rounds, as I think there are over 100 people signed up for it, as mentioned, is over 30 countries. Just to give a little bit of a flow process today, we're also going to accept questions in each of 3 different sessions. with Jean-Paul going first, and then as I understand it, Sarah will go next, and then Sabrina will go last. But at the same time, there could be opportunities for overlap. And now we get a chance to participate in your great work, Jean-Paul and team. Did you want to? And I would say there's many other co-authors for your work, which aren't able to be highlighted today, but they are definitely, let's just say, brothers and sisters in the phage community, and will also equally be celebrated, I hope. So, are we good on your end from a technical perspective?
Yeah.
Great. So I see you're sharing screen, and let's let the real fun begin.
Wait, because
Okay, I see your screen.
yeah, but I cannot see it. That's a bit the problem. I needed to see my slides. Okay, I see it now. Okay, so many thanks for the introduction. And good morning to everyone in Canada. And thank you for having me. Um, we were asked to present the framework that we have for phage therapy in Belgium, and with an emphasis on the publication of the first 100 phage therapy cases. So I will start with a more general overview, explaining why we developed this personalized phage therapy framework, um, and also explain what it is precisely. So first I need to, and this is new, it's one of the first times I need to do this. I have to present a disclosure slide. So the views expressed are those of the presenter, so me. Uh, but the content has been reviewed, though. I don't know why, but. And then, of course, I have no conflicts of interest, and this presentation is intended solely for educational and scientific exchange. Um, we began our phage therapy research in 2003. And this slide is mainly to explain why in my presentations I often refer to science fiction. So when we started in 2003, of course, we needed funding and we had to apply um, to the Royal Military Academy. And you can see the scoring sheet of all the proposals submitted in 2003. So you can see 26 proposals evaluated by many experts. Um, 11 got funded, and we were at the bottom. We scored 8 out of 20, got no funding. We had a military angle, bacteriophages against bacterial bioweapons, but still, this was not enough. And one of the experts had written on the proposal on the first page that it was science fiction. Now, everything is relative because the proposal scoring 7 out of 20, just below us, was about collecting postcards featuring military uniforms pre-Waterloo. So it's even 1 century older than the phage therapy, which was first applied in 1919. But I'm happy to say that today, but more than 20 years later, we have developed a phage therapy research um, framework, starting from isolation of phages in the environment to the application of these phages in patients. Of course, this is in collaboration with many research institutes. The most important one is the Eliav Institute. So from the beginning, we decided not to reinvent phage therapy. So our starting point was the 100 years of experience at the Eliav Institute. As most of you know, phages have been classified as medicinal products or drugs in the US. But you all know that they are very different from conventional drugs such as antibiotics or aspirin. I will just quickly discuss two characteristics that are important with regard to phage therapy. First one is selectivity. So antibiotics, they usually target many species of bacteria, or at least they used to do. And while bacteriophages, they only target a part of one bacterial species. Advantage, of course, is that we can target the pathogens and spare the beneficial commensals. Big disadvantage is that we need a handful to dozens of phages to target only one bacterial species. Here you can see an analysis of the phage therapy requests in Brussels and San Diego. And from both pie charts, one can conclude that we need phages against more than 30 bacterial species that were involved in the phage therapy requests at both centers. You can see pie charts not identical. There will be geographical variations. And I'm sure if we redo this analysis today, also pie charts will be different with some emerging species like Klebsiella pneumoniae or Providencia stewarti, and also different strains that are emerging. So this means that we need hundreds of phages. Then there is resistance, because of course also bacteria can develop resistance to phage. Here is a picture with all anti-phage defense systems that were known in 2023. You can see that CRISPR-Cas is there. This was the first one that was discovered. It's also the most renowned one. But almost every month there's a new one that is discovered. Of course, these impact phage therapy, but we have no idea how these anti-phage defense systems will be selected or not. Will they persist? Will they spread? We have no idea. And if they do so, they might create problems for us to select the right phages for therapy. We know that once we have selected the phage that can infect the patient's strain and will not have any major anti-phage defense systems against that phage, then what will happen is that during therapy, resistance will be selected, but what will be selected is mutants with alterations in the phage receptors. So we never see any anti-phage defense systems showing up during therapy, but we see mostly mutations in receptors of bacteria. Sabrina will go more into depth into these resistance mechanisms that show up during therapy. Now, I've explained these issues of the selectivity and resistance, but still in Europe and I think pretty much everywhere in the world, the regulator or the policymakers consider that the existing regulatory framework is adequate for phage therapy. So this is the example of Europe where the Commission has formally stated this. So this means that we need to follow the conventional licensing pathways. So it means manufactured according to good manufacturing practices or GMP, preclinical studies, phase 1, 2, and 3 clinical trials, and then one can apply for marketing authorization. Now, of course, this requires a lot of time and money. Someone calculated exactly how much time and money on average. And of course, over time, let's say over the last 30 years, companies have tried to follow this conventional pathway. But today, as you all know, we have no phage medicines on the EU or US markets. And this is an interesting paper in Nature Biotechnology in 2004. And it contains this table with phage therapy companies or companies that try to market phage therapeutics. In 2004, you can see 11 companies. So you see, founded between 1994 and 2003. None of them exist today. Only two survived, Gangagen and Intralytics. But both initially focused on agro-food applications. And now also Intralytics is moving towards the human phage therapy field. What is the situation in 2024? So I looked in the databases where clinical trials are registered. And I found 25 randomized controlled trials with natural phages. So this is not about phages used as vectors for payloads. So this is real natural phages. 25 clinical trials, 7 completed, but as you all know, without convincing results. 3 terminated by the company, and 15 are either active recruiting or have an unknown status. If you can see, most of them, all of them except one in phase 1/2. I think there was one phase 2/3, but also there were no interesting results. But importantly, you can see that they target 7 bacterial species. And 71% of them target only 2 species, Staphylococcus aureus and Pseudomonas aeruginosa. And as I showed, if we really want to have phage therapy for all patients who need this, we will need phages against more than 30 bacterial species. Also, a limited number of indications. These are mostly commercially viable indications. So you see what is in the pipeline only targets commercially viable species and indications. So even if all these products would come on the market tomorrow, these will not solve the MR crisis. Already today, 1 year later, 4 of these studies have been discontinued. And we need phages today. So that's why in Belgium, we decided to develop a pragmatic framework. And I think this is an important slide. So you can see here Alain Fauconnier from the competent authorities of medicines, let's say the Belgian FDA. And he came up with 3 possibilities for phage therapy. And one of them was magister preparations. We opted for that solution, magister preparations. And even though let's say the Belgian FDA was quite okay with that concept, they couldn't do much because they are not making frameworks, they implement them. So we decided to look for policymakers that could help us. And here you can see in the yellow box the member of the European Parliament, Michèle Rivasi, who was really an advocate of phage therapy. And she sadly passed away last year. And she was member of the Green Party, and she activated the Green Party in Belgium, and also its partner, the Socialist Party. And they started asking questions. in the Belgian parliament. And you can see Dr. Maggie de Brock, who was then the Minister of Public Health. So we were lucky it was a medical doctor. She was confronted with questions from the Green and Socialist Party about the status of phage therapy in Belgium and why don't we have phage therapy in Belgium. And then she answered, black on white, that she was aware of the problems getting phages through the conventional framework, and she opened the door to magistral preparations. Very importantly, she asked the competent authorities for medicines to help us setting up a framework. And this gave a mandate to Alain Fauconnier, but also to Peter Jan Sijsens from Cienzano to help create this framework. And this is the framework. So we have a phage bank with characterized phages. From this bank, we can select phages to be produced as phage ingredients. So they are called active pharmaceutical ingredients. And these phage ingredients need to be produced and also quality tested according to a monograph. And also important is that we cannot do our own quality testing. This needs to be performed by a Belgian approved laboratory, a certified laboratory. And then these phages, together with the certificates, can go to the hospital pharmacies, where they can be integrated into a magistral phage preparation upon prescription by a physician. So at the pharmacy, different phages can be mixed in a carrier, which can, for instance, be an hydrogel for burn wound treatment, or it can be an IV solution or whatever. Then this preparation can go to the patient. And I need to stress that these phages are not GMP certified, and there were no preclinical or clinical trials. And there's also no marketing authorization. This doesn't mean that there is no quality. So, of course, we have our standard operating procedures. We work in clean rooms, and you can see on the bottom right 2 examples of magistral phage preparations. This is the site from Cienzano, which is the Belgian approved laboratory that liberates the phages for therapeutic use. You can see, so they are responsible for the safety of therapeutic bacteriophage preparations. And it states that the certificate produced by Cienzano allows the phage to be used in pharmaceutical preparations. This is what is tested. So on first genetic analysis to confirm the phage is strictly lytic, that there are no known toxin or antibiotic resistance genetic determinants, and also a screening of the production host. And then for every phage ingredient batch, phage identity will be determined, phage titer, pH, bioburden, endotoxin level using the LAL test. But somewhere this year, this LAL test will be changed, and it will be the MET test that will be used, which is broader. So it doesn't only measure endotoxins. Also, we will have a prophage threshold. So we are allowed to have prophages in our phage preparation, but up till now there was no limit, so there will be a limit. It will also state that these prophages can, of course, not contain any toxins, etc. And we can train or pre-adapt phages without this phage to be considered a new phage, requiring a new genetic analysis. But of course, also there we will have a threshold because you cannot adapt indefinitely, because in the end you will indeed have another phage, I think. Of course, very important also to have information on the stability of the phage when this phage goes to the pharmacy. So stability in different carriers, for instance, IV solutions or gels for wounds. Also know the stability when it's nebulized. And since the end of 2024, this system was also implemented in Portugal. Now, maybe you would like to know how much this costs, huh? How much do phage APIs cost? Well, I made a quick calculation. So luckily we have an analytical accountancy in the military hospital, so everything is included in the price, from the printed paper to the roadworks nearby the military hospital. So you have personnel costs, the clean room zone, equipment, reagents, disposables, operating costs, quality control, etc. Even telephone costs. Uh, and I can tell that the production of one phage API batch costs a bit more than 11,000 euro. 14,000 if it's a new phage, because then we need to add the cost price of the genetic passport. Now, what does it cost per application of an API, phage API? Well, if the entire batch would be used, it would be very cheap, about 50 euro. But in reality, this never happens. Huh? So in reality, it will cost 888 euros for a phage that is used regularly, to 14,208 euros for a phage that has been used only one time in one patient before it expired. On average, it's 3,489 euros per phage application. And everything is financed by Belgian Defense. Now, come to Arrowsmith's dilemma, and it's an old dilemma. It dates from the 1930s. I don't know if you all know the book Arrowsmith, written by Sinclair Lewis in 1926. It got the Pulitzer Prize, was turned into a movie, successful movie. And it's about Martin Arrowsmith, who discovers a phage that destroys Yersinia pestis, which of course causes the bubonic plague. Now, this book and movie are very realistic because they are based on, like, let's say, the advice of a famous bacteriologist, which is called de Kruif. And he witnessed the isolation of the first Staph aureus phage in New York. So his advice was used to create the character Martin Arrowsmith. And in the movie and the book, Martin Arrowsmith needs to choose between pursuing scientific precision through randomized controlled trials, or pursuing the immediate welfare of a population threatened by the plague. So he needs to choose. So you see here, this is a picture from the movie where you can see Martin on the right, and he's ordered to perform a randomized controlled trial by his hierarchy. So he doesn't want to, but he's really forced to do so. So he goes to a Caribbean island where every year there's a seasonal outbreak of the plague, and he gives phages prophylactically to half of the population and water to the other half. And it's randomized, of course. And then the plague arrives, and patients that got water start to die, while the patients that got the phage survive. Unfortunately, several of his colleagues, but also his wife, didn't get the phage, and they die. And then he decides to give phages to the entire population. So this is just to illustrate that I think it's easy to decide these things when you are in your office and to say, okay, you will not give the phage to half of the patients. But in practice, I think in the field, when you work in hospital, it's very difficult to deny a possible therapy to patients. So that's why we decided to do both, because of course it's our obligation to help persons in need. So this is yeah, the paper about the 100 consecutive cases, just to show that we didn't focus on commercially viable indications, so we targeted all possible infection types. We targeted 14 bacterial species, which is already double of what's in the pipeline today. And we targeted sometimes 3 species in one patient. For this, we needed combinations of 26 phages, 13 of which were pre-adapted and often used in combination with antibiotics. Now we are now over the 200 cases, and I can tell you that we have produced more than 80 batches of more than 40 phages for these 200 cases. So imagine doing this GMP, having preclinicals and clinical trials. This pre-adaptation of phage training is not the correct word. Of course, we know that this can increase pathogen clearance and, very important, also lower resistance evolution. We also start to know what this training does. It mainly mutates the genes related to the tail fibers of the phage, so where the bacterium becomes resistant by mutating the receptor for the phage can become more effective by mutating the tail fiber that connects to the receptor. It makes sense. Results will be discussed by Sarah. She will also present a few cases more in detail. Told that we decided to do both, so to treat patients, but also to get involved into clinical trials. So this is one that is running. I think about 30 patients enrolled now. The Phage Force study, you can find the protocol in the open access publication. And this is the efficacy study. It's a multi-centric study which started a few weeks ago. And we also provided phages to help our Ukrainian colleagues, but also there, Sabrina will have some more information. So, of course, when we want to bring this personalized phage therapy concept to a larger scale, we are confronted, of course, with implementation challenges. I have no time to go into detail, but we recently published a letter about these challenges. One of them I can discuss. It's of course the logistics of personalized phage therapy. So all these lines are shippings of phages, bacteria, sometimes patients. For every line, you need to deal with the shipper, with customs, with regulators, sometimes with insurance companies. It's a lot of work. So that's why our research now focuses on what has been called a phage printer. So we would have a device, we would introduce a patient sample, you would get DNA extraction, sequencing of this DNA, and then AI would find the genome of the bacterium or bacteria that are involved in the infection and would predict the phage genome that would target these bacteria. And then using synthetic biology processes, we would produce this phage synthetically. So we would not produce a synthetic phage, but we would produce a natural phage in a synthetic way. Then we hope to get personalized phage preparations. So it would be an instant point of care synthetic production of phages. Yeah, we recently published some conceptual schematic of such a machine. So if you want more details, you can look up this publication. Technically, we can do this today, but this is the equipment we would need. And it would take a lot of time. It would cost a lot of money. The synthesis of a phage genome alone would cost you more than 20,000 euro. But this is the first computer. And today we all have a smartphone that is, of course, much smaller, uh, has much more capacity than this first computer, is quite cheap, relatively cheap. I would like to make some publicity for the study group for non-traditional antibacterial therapy, Scenta. A lot of phage therapy people there. Then I would like to thank my colleagues and collaborators, of course, Sarah, Patrick, and Gwenaël of the Phage Therapy Coordination Center, of course, the Eliave Institute, KU Leuven, University of Ghent, and of course the phage therapy providers in more than 30 hospitals. And then I would like to thank you for the attention.
Great, thank you so much. Ran and Gina, time for a quick question or discussion as we go into the next talk.
Just a provocative question. When do you think your suggested machine will be available to use by all?
In the paper, I predicted 2035. I have no idea. It will depend on the evolutions in different fields. Already there are some recent evolutions in the synthesis of genomes. When I wrote this paper, I think it was limited to a few thousand BP. There are now two companies that offer 20,000 BP in one stretch. So it's evolving quite quickly. The cell-free production system that you also might know that it's being developed in Munich also is evolving nicely. In the beginning, it was limited to E. coli, but now Klebsiella, some Pseudomonas, even some gram-positives. So it will depend on all these developments. Also AI, there are a few papers on prediction of phages using AI. So I think technically, if a big company gets interested, for me it could be 2035, but it will really depend on whether there will be like important players interested.
Okay, very good. We'll now transition over to Dr. Sarah Djebara. And almost there.
Almost. Yeah. Is this okay?
Fantastic.
Perfect. So thank you. I would try to talk about the clinical aspect of this Belgian phage therapy experience. So I will try to talk a little bit about the 100 case paper, but I will focus on two clinical case to be more practical. The same disclaimer and disclosure than Jean-Paul. Voilà. So just to come back a little bit on this paper, and I will quickly summarize it because I would like to talk about two clinical case, and I think concretely it's more interesting, I would say. But so yeah, so you know that this paper we talked about the 100 consecutive patient who had been treated with phage therapy. We had to use, as Jean-Paul mentioned already, 26 phage in order to treat 100 patients. 13 had to be pre-adapted, and we used 6 phage cocktails. We used phages together with antibiotic in 69%, and we know that we had 70%, you have 70% less probability of microbial eradication if you were not using phages together with antibiotic with an odds ratio of 0.3. We targeted 14 different bacterial species, and among them, the 2 most important ones are Pseudomonas aeruginosa and Staphylococcus aureus, also because these are the 2 bacteria for which we have the most important number of phages, even if I will show you how much phages we have in our collection today. So we were able to see 77% clinical improvement and 61% of pathogen eradication. We were able to assess, but not in all the treatment, the possibility of a phage and antibiotic synergy, dyssynergy antagonism. Just to update you that now we are doing this systematically, but before it was not the case, also because now we have an Omnilog system which is helping us a lot. But among the tested, 9 out of 10 show synergy between antibiotic and phages. And we were also able to assess phage immune neutralization in 13 patients. And 5 out of 13 showed serum neutralization antibody. But despite this fact, in the majority of the case, it didn't hamper the good clinical outcome of the patient. So I think we will discuss further on what is, to our opinion, the reality and the meaning of those serum neutralization and why we are still measuring it. And also, of course, but Sabrina also will talk about this, the phage resistance emergence. We were able to assess this in 16 patients, and 7 out of these 16 patients showed resistance emergence. But again, it didn't hamper the good clinical outcome in the majority of the cases. Adverse events. So we had 7 non-serious adverse events, which can go from a little bit of fever, a little bit of redness when you have a local administration, and also sometimes a little bit of cough for nebulization, but nothing serious up to now. So we gave phages and we worked with 35 different hospitals in 12 countries and 29 cities. So as Jean-Paul is mentioning, logistically, it might be a hell sometime. So here, just to summarize, but I think you have already see that, as I said, we targeted 14 different bacterial species. And also, this is on the left, you have the distribution of the different legal approach. So, of course, since 2018, we have the majority for Belgium. The legal approach is the magisterial framework, of course, but it's different in different countries where we are working. You have the different indications. The most important one is low respiratory tract infection, and I will show you some figure for the 200 consecutive treatment, and you will see that it's not changing actually. So it's always low respiratory tract infection, skin soft tissue infection, and musculoskeletal infection. And here is the distribution of age. So the medium age is more or less 50, I would say, but you can see that we are even treating very young patient and sometimes very old patient too. Okay, so of course there is limitation of this study. You know that we had very different indication and different route of administration. There is no control, no blinding, no randomization. The evaluation of the efficacy and safety is based on non-standardized, I would say, test, but on subjective judgment of the physician. So it's important to say that. And unfortunately, we were not able to get samples for all those 100 patients we treated. So this is why we are advocating also for phage and to have the capacity to collect the data in order to have a better control and idea of what we are doing with those phage treated patients. So phage reality in the clinic. So I'm sorry for the next picture because it might shock some people. So I'm very sorry, but So you know that at the Queen Astrid Military Hospital, we are receiving a severely burn wound patient, but also, as you know, that in 2016 there was the bombing attack in Brussels. We also received this kind of blast injuries and also bombing wounds, and you can easily understand that they were really prone to have MDR, but difficult to treat infection, I would say generally, but also MDR bacterial infection, of course. So I will talk about two cases to illustrate some important, I would say, phage concepts that we are using on a daily basis. So the first one is the story of a young 12-year-old boy who suffered from a very important necrotizing fasciitis caused by PVL producing MRSA. So the story is that 8 days before his admission to the ICU, he had a very minor trauma with superficial scratches. I think he was playing football. So superficial scratches on his ankle, on his left ankle, and that very quickly got infected. So he had like very quickly after 48 hours, he started to get his leg was swollen, and also he had some confusion, some fever. And he was really suffering from the swollen and the redness of this leg. So he was admitted at the ICU where he had, of course, empirical antibiotic after hemoculture, of course, all the sampling and also the sampling on his leg, because there was already passes during his first surgery, because he had to go on a surgery because he had a compartmental syndrome, actually. And he was put under cefotaxime and flucloclacilline and clindamycin. But despite this and the ICU care, he was really in a very septic shock with multiple organ failure, with embolism with septic embolism in the lung, with the pleural and emphysema, but also effusion in the peritoneal cavity that was infected. All of this because he had actually a mitral valve infection. So when the hemoculture turned to be positive for MRSA, he got a switch on his antibiotic, of course. So he continued the clindamycin and the vancomycin. But despite this, and despite the first surgery, unfortunately, the septic shock got worse with multiple organ failure, renal insufficiency, hemodynamic instability. And this is why we were asked to give phages together with antibiotics. So we started to give phages, but what is original here is that we gave it in a very with different way of administration and for different bacteria, for different bacteria species. So we started with intravenous treatment of phages against Staphylococcus aureus with ISP phage. He received also the phages through nebulization because he was also suffering from pneumonia. Also, we received phages through the pleural cavity and also the intraperitoneal cavity through drains. And also, of course, during the dressing of his leg, he received the phages. And after 12 hours of the start of the phages, he received also a ceftaroline, so this fifth generation cephalosporin. And after 40 hours, he didn't have anymore any hemoculture positive, which was already a good start. And he started slowly to recover from his septic shock. So everything went better. We were able to remove the drain in the pleural cavity and also in the peritoneal cavity. So everything went better, but unfortunately, the leg was really still in a very bad condition. So he had to go for a flap surgery, and he received phages topically. At that time, he was also suffering from a Pseudomonas aeruginosa infection, so he got a switch to meropenem, and he also received phages against Pseudomonas during the surgery. But despite of that, unfortunately, he had to go for an amputation. We were not able to save his leg. He also had a valve surgery due to the mitral valve endocarditis. But after that, he was feeling much better. Also, last but not least, he was also infected with Stenotrophomonas maltophilia by the time he had this amputation. So he also received phages against Staphylococcus aureus, Pseudomonas aeruginosa, and Stenotrophomonas maltophilia at the same time. And after that, he didn't have any more in his sampling any more of these three bacteria, and all the hemoculture, like I said, were still negative. And he was finally discharged almost 2 months after the beginning of his stay at the ICU. So just to show this to you, but I think that Sabrina will talk very much longer than I will about this. But what was interesting in this case is that we were able to isolate when we were doing the bacteriological sampling during the follow-up of this patient during his stay in the ICU. He had some Pseudomonas aeruginosa resistant mutant, despite the fact that he received at the same time 3 different phages with using 3 different receptor at the same time. So, for example, phage PT07 was using most probably a part of the efflux pump as a receptor, and we were able to find this mutation and also DNA GRAS mutation. This double mutation were shown to recover the sensitivity of 2-fluoroquinolone with a decrease in the MIC of ciprofloxacin from 4 to 0.5. Another phage is PNM, which is known to use type 4 pili here as a receptor. And phage 14.1 was using LPS, so you can see that those phages were using part of virulence factor of the bacteria as a receptor. So the question was, okay, was there a virulence, a reduction of the virulence capacity of this bacteria? So we used the Galeria melonella model to check this, and indeed you can see that the probability of survival of the bacteria decreased in those phage-resistant mutant compared to the original one. So this, together with the fact that we were able to check for the synergy between the antibiotic and the phage, you can see here the green line here is really showing an inhibition of the growth of the bacteria for more than 72 hours when they are used together, phage and antibiotic. And when you compare here, the yellow line is the antibiotic ceftaroline alone. So you see that there is a clear synergy effect between ISP and ceftaroline antibiotic. So this probably made our conclusion that the patient clearly had a recovery thanks to the combined effect of the phages and the antibiotic, but also, as you can see, the resensitization and the reduction of virulence in some phage-resistant mutant, which is very interesting. Another case I want to share with you is the case of the 30-year-old lady who was unfortunately a victim of the bombing attack that happened in Brussels in 2016. So she was actually, I can say her name because she's really clearly advocating for a lot of things, and she's a very courageous and amazing woman. It's Karen Northschild, and she's a US athlete actually. So she was at the airport when this happened, unfortunately, to her. And the story is that when she arrived at Erasmus Hospital in Brussels, she had a cardiac arrest due to the constant bleeding. And of course, she had to go for a quick surgery where they removed a part of the iliac bone. They had to amputate the iliac bone partly, and also to put some fixator on her left hip. And a few days after, unfortunately, this fixator and this surgery, she got infected first with Enterococcus faecium, with Pseudomonas aeruginosa, and also Enterobacter cloacae, and Klebsiella pneumoniae was already present at the beginning. So, of course, she received a lot of antibiotic treatment. She had a lot of secondary effects, of course, due to amikacin, the deafness, tubulopathy due to colistin, and also she had neutropenia, febrile neutropenia due to meropenem. So she was really suffering also from the antibiotic treatment, but of course there was no choice. And they wanted to put an autograft because the defect was very important on her femur and hip, but unfortunately it got severely infected with the sinus tract, also with discharge of pusses, with a lot of pusses. And we were able to see that there were two Klebsiella pneumoniae isolate that were XDR, unfortunately. So they were a bit stuck because she was not evolving in the correct way. So we were asked to give phages. At that time, we didn't have in our collection phages against Klebsiella, so we asked the help of Elviva Institute. So they find phage M1. That could be used against those two isolate that were found in the pusses. And we needed to make some pre-adaptation. So as Jean-Paul told you, the idea is to have a better lytic activity with a low emergence of mutant, resistant mutant, for at least 24 to 48 hours with a low MOI, less than 1. So after 15 rounds of coevolution, thanks to the Appelman's method, we had a good phage that could be used for the therapy. But unfortunately, despite the fact that the ethical committee said that it was okay to use it, unfortunately, the medical team was not really happy to use phages, and the patient had to wait actually almost 2 years before receiving. So she was in a revalidation center, and she had to wait for almost 2 years before the use of the phage, because actually nothing was improving for her, and despite all the antibiotic treatment, as I said before. And finally, the Klebsiella turned out to be PON drug resistant. And when we tested again, so almost after 2 years, the phage that we adapted on the first isolate, we were very lucky because finally the phage was still active. So it was still possible to use this phage, sorry. And the phage was finally used during a surgery, and together also with ceftazidime avibactam and moxifloxacin as well. And finally, the patient recovered. So I think that you can see here the picture before and after the treatment, and also the consolidation on the X-ray and on the CT scan, which was positive, of course, for this patient. So when we analyzed what happened with this patient, so first of all, we analyzed the synergy between phage and antibiotic, and we were able to find this phage between ceftazidime avibactam and also with meropenem, but not with colistin. And also, what was also interesting is that we check in vitro also on a model of a biofilm. We tested phages. So I think it's 7-day mature biofilm, and we gave phages on 3 days consecutively, together or alone or together with antibiotic, and you see that the best reduction of the bacterial growth is obtained with phages together with antibiotic, which was also something very interesting. It's just to show you here the phage pre-adaptation. I told you about it, and as Jean-Paul said, we had this missense mutation here on the distal tail fiber protein that might explain a better interaction between the phage and the bacteria, that could explain the better activity. But of course, to check this, we should have take this mutation and put it in the original phage, but of course we were not able to see it, to be sure that the association between phenotype and genotype is in concordance, but we are imagining that this is the case. When we are adapting strain, of course, we are sometimes reducing the host range. It's a risk, but recently we were in the Congress, and the very interesting work of Joanna, I think that she showed something very interesting, because she was testing and trying to adapt and to see that when she was using a biofilm model for cystic fibrosis patient, and she were using different isolates coming from different sputum, she were able actually to increase the host range on this biofilm model with those different sputum. So I think it's quite interesting for the future, and maybe what we should do in chronic wound or chronic infected patients. So phage adaptation, this is something that we are regularly doing also to adapt our own phage production and to increase their activity, and sometimes to adapt on a specific patient. And phage immune neutralization. So for this patient, it appeared after, I think, 18 days that she had the phage immune neutralization antibody. Of course, it will depend on the immune status of the patient, on the phage, of course, and on the way you are giving the phages. But it's not always the case, because before we were always saying, okay, this is happening more often when we are giving phages intravenously. But here, this is the work of people in Spain, and they showed that neutralizing antibody in the serum after nebulized phage is also happening actually very quickly after phage treatment. But in this paper, and also in our experience, it didn't hamper actually the outcome. But the thing is that it's interesting still to measure it, also because you might need to use those phages in the future, and maybe you need to change, or maybe it can explain sometime a mitigate result. So I think it's still important to document it. And also because we are treating patient now, I would come to this on a chronic way, actually. Some cystic fibrosis patient, for example, or other chronic condition. So this is the available phage API for the moment, but actually we have more, total of 123 phages in our collection. So just to show you very quickly, 200 phage treatment now and 183 patients. It's a little bit more now. Just the distribution, geographic distribution. So you see that Portugal and France are the followed country we are working with. And of course, with the approach that Portugal adopted on November, I guess it's going to be even more. This is the number of phage treatments per year. Also, something very interesting that we showed already on our paper in 2019 is that we are not only treating MDR or XDR bacteria, we are treating difficult to treat infection. And as you can see, actually, the majority is non-MDR bacteria. This is the outcome, but actually we cannot say a lot of things because it's really preliminary and we are missing a lot of information. And this is the outcome and microbial eradication depending on the different indication. Different bacteria species, you see that now the third one is Mycobacteria abscessus. So in conclusion, I think that our experience showed that definitely the personalized approach is taking a lot of time, is complex, but is definitely the best. Also because we know that unlicensed phage preparation is more efficient and less prompt to spread the phage resistance in the phage than phage cocktail, of course. I think that now we came to a point that we definitely need to try to fix some clinical guideline and learn as we go process with for some indication, like I would say for musculoskeletal infection and maybe for respiratory condition. I think now we need to have phage hubs and working group like we are doing here now. GCPR is a very good approach. We are very encouraging everyone to use the phage history because we really need to document better our cases, definitely. Thank you. And yeah, I want to thank, of course, all the lab, Jean-Paul, Maya, Joanne, Christelle, everyone, Gwenaël, and Anandi also, and all the people from the academic and hospital, and of course, patients that are trusting us. Thank you.
Thank you very much. Gina Aran, yeah, seeing some clapping there. Fantastic. We're almost like having an opera of phages right now with everyone and their presentations. Any quick questions from Gina Aran before we give it to Sabrina Green?
This may not be a quick question, Sarah, but I am interested in the process for deciding on a certain regimen for each individual patient. Do you, at Queen Astrid, do you review each case and recommend dose, duration, frequency for each case?
Yes.
Okay.
So as Jean-Paul said, we are using of course, the experience of Eliava. This is the beginning of I would say our experience. Of course, it's their experience, thanks to them. But we are adapting in our practice. And also, I think this is like a flexible model to talk, because I want everybody to feel comfortable, which means the surgeons or, if they don't want to put drains or if they feel more to do this or that. So of course, it's not like a systematic approach for everyone, but it's really, personalized. But yes, I'm reviewing every treatment and every case.
Fantastic work. I'm very much interested in anaerobic phage therapy as well, and the B. fragilis in particular didn't work out. But as we looked for C. difficile or other challenges, maybe if we have a little bit of time afterwards, we can think about how we would look to approach those anaerobes. I think there's a few strategies. So I do believe we should transition to Dr. Sabrina Green, and we are also a pretty strict timekeepers. I'm sorry we have to finish in about 22 minutes, but I hope that will be sufficient time to both hear Dr. Green as well as have some comments. I do like some extra comments in the chat as well as we're going through, and please, as we work through this, take the time to fill out the evaluation form. Dr. Green, Sabrina, please. I think we can share your screen. Great. And the floor is yours.
Okay, you see everything?
We see everything.
Great. So some things will be repeated. I'll try to go quickly, but they say if you repeat it 3 times, you really remember it. So I think that's good. So I wanted to first define the AMR problem. My slide is a bit censored from a previous presentation I made, but this pretty much sums it up. We're AMR is really bad. It's only getting worse. I have been working here in Belgium only the past few years, but they have been working on this for 2 decades, developing this phage therapy process. And of course, it's led to this big paper, this 100 cases paper that we've been discussing. So I'm in KU Leuven right now, and we focus more on sequencing and genomics and understanding what is happening after phage therapy. So for us, we got to play around with some of the samples that we got after treatments. So among these 100 consecutive cases, 23 cases had enough samples in order to assess resistance to phage. 7 were already, or 7 were cases had resistance phenotype among these 25, and 2 were published already. And so we got to assess 5 cases, and this was 21 samples of Pseudomonas aeruginosa. So as I said, we perform sequencing. We do Illumina and nanopore sequencing. We do these hybrid assemblies. Here is just kind of an overview of the entire genome of this bacteria from this patient. The green circle indicates that the bacteria was sensitive to the phage, and then the pink circles indicate that they were resistant isolates that were subsequently sampled after treatment. So as Jean-Paul mentioned, we only see changes in the receptor when we get resistant isolates. So this is a good example. You have a phage that's well characterized, PNM, many papers about it. It's clear that it uses type 4 pili as a receptor. And so what happens when you have a patient that's treated with this phage, you see resistors and you sequence them and you find mutations in this region that's important for the type 4 pilus. In this case, it was an amino acid change in pill C. This is the inner membrane component of this type 4 pilus. What happens in another case where, again, they used PNM. Here we see a mutation, but in another gene, in pill R. This is part of a 2-component system that's also important for this type 4 pilus. So the type 4 pili, it's a virulence factor for Pseudomonas aeruginosa. So naturally, we think, do these mutations lead to a cost to virulence? For that, we use the Galeria melanella model. It's a wax moth larvae assay. And what we find is when we assess this, in this case, it was the patient that had an isolate that was resistant and had a mutation in pill C, that more or less the Galeria die within 12 to 18 hours. So we don't see a significant reduction in mortality. If we want to kind of associate this to the actual patient outcome, you can see that this patient number 30 did not improve clinically. So maybe, yeah, you had this change, a resistant change, but there was no cost to virulence and the bacteria remained virulent. Then you have the case where patient 54 that had this mutation in pill R. And here, again, the Galeria die with within 18 hours. There was a slight reduction in mortality rates in the isolate that had the mutation. And this patient did improve clinically. So I don't know if this actually means anything, if there is a virulence defect in this resistor, but it's something interesting that I think we can look into more. And as Sarah described, and she went through this case very nicely, but I just want to emphasize again that these were 3 phages targeting 3 different receptors. So in the literature, you often hear this, you use 3 phages or multiple cocktail of phages targeting different receptors, you will not get resistance. But yes, we see resistance to every phage. So we see in this case, we had a phage PNM, again, that uses type 4 pilus. We have a mutation in FIMV, a major deletion in this region. We have a phage called 14-1, also really well characterized. You have several deletions in this inner core component of lipopolysaccharide. And finally, we have a phage PT07, which is less characterized, but when you search this genome for similar phages, you see that there are phages that are very similar that have been well characterized, and they use MEX-XY-OPRM as a receptor. So in this case, for this resistor, we saw this SNP in this region, MEX-B. This is the inner membrane component of the efflux pump. And so what we also saw was a mutation in gyrase A. So this is within a region that's, fluoroquinolone determining region, quinolone resistance determining region. So if you look up old papers about this specific region and you make these mutations, you see these papers where they made this exact same mutation in this exact same amino acid, but the other way around, that's how you see resistance to fluoroquinolones. And so it is the combination, I think, of these 2 mutations that then we see this decrease in MIC, as Sarah described, from greater than 4 micrograms per mil to 0.5 micrograms per mil in ciprofloxacin, and greater than 8 to 1 microgram per mil in levofloxacin. So you have mutations, but then you have sensitivity to antibiotics. This is good. Now, are these mutations also leading to a cost to virulence? Again, we assess this in the Galeria melanella model. And what we saw is that these Galeria, they survive 48 hours, possibly longer, but we stopped the assay. And that is only if they have a combination of mutations in all of these regions. So you really, if you have mutations and major mutations in these regions, naturally the bacteria, it's going to come to a cost, at a cost to the bacteria. So I'm not sure if these cases were described, but patient 82 and patient 64, you see this when we sequence the resistors, they're actually a different sequence type. So they're not even the original bacteria, but they're still Pseudomonas. And so, yeah, if your phage is specific for a bacteria, and you have another Pseudomonas that comes up that the phage cannot hit naturally, it's resistant, it's not its host. These patients did eventually improve clinically. And again, I'm just making assumptions that probably why they eventually improved clinically is because, you know, it's the combination of the antibiotics and the phage that are really necessary because phage are so specific. You really need something there to hit the other things that may come up. So I used to show this make antibiotics great again, but I think it's a bad time to do that. So I changed my slide a bit. Say phage and antibiotic are stronger together. Very European Union solidarity phrasing. And I'm just going to use a little bit of time to talk about our project in Ukraine, if that's okay. So AMR, of course, is getting worse, and Ukraine is you know, having to deal with this. The clinicians are in the middle of this. They're having to deal with antibiotic resistance. These patients are being spread around the world. It's further spreading this, and yeah, it's getting worse, right? And so we started Phage Support Ukraine. This is mainly led by the Queen Astrid Military Hospital to provide phages, but also training and an infrastructure in order for them to produce these phages and treat patients there. We received these isolates and we sequenced them, and naturally what you would most assume if you were a clinician or working in this field, that the most predominant and antibiotic resistant was Klebsiella pneumoniae. You can see in this antibiogram that these strains are more or less resistant to everything except colistin for now. What we find when we sequence these strains, not only directly from Ukraine, but from patients that come from Belgium, Latvia, and Germany that were displaced due to the war, that they more or less fall into two sequence types, two major groups, ST23 and ST395. ST23, actually, it's a clonal group within it that is a bit distinct from ST23, but I won't go into that. But you can see from this tree that they are carrying lots of virulence factors, lots of antibiotic resistance genes. And how do they carry this? They carry this in huge plasmids, 200 to 300 kb plasmids. And this is just an example of one from one of these ST23 strains. It's carrying NDM1. It's carrying Arabactin, this siderophore virulence factor. It's carrying ARM-A, which provides resistance to aminoglycosides. So it's carrying a lot in these huge plasmids. So working with the Queen Astrid Military Hospital, of course, Maya provided us or found these phages that could target many of these strains. Not only the ones that come directly from Ukraine, but the ones from patients that are displaced in Belgium and Germany and Latvia. They've all been sequenced and characterized. Some have been named. And we perform growth curves to see how effective they are. Of course, with Klebsiella, you always see resistance, but it's a matter of how long you can tamper this resistance. And so, yeah, we see there's high potential for these phages. And I just wanted to talk a little bit about the assessment of resistance, because that's sort of the theme of my presentation a bit, is that when we do in vitro assessment of resistance of some of these phages on these strains, it's kind of a way to see what the potential receptor is of the phage. If you see a SNP in a region, it could be that the phage needs this, and so then it changes, the bacteria changes. And so we get an idea of what receptors these phages are using. In this case, it's more or less what you would think, lipopolysaccharide, capsule, biosynthesis, some outer membrane proteins. And what we also find that there are large deletions in these regions, sometimes 10 kb, 6 kb. The most interesting we found was this strain completely lost this 350 kb plasmid, which I found very interesting. And so we assessed this phage was named Mavka, sorry. And we assessed this, and we found that in 3 out of 9 biological replicates, we see the complete loss of this plasmid when this bacteria becomes resistant to this phage. And when it doesn't lose the plasmid, we see mutations in this plasmid. And so this suggests that this phage needs something in this plasmid, whether it's a receptor or something else that is occurring. And so that is giving this plasmid dependency. And so what does this strain look like? Again, I showed you the antibiogram of that strain, and now we see, yeah, this similar kind of trend that we saw in the 100 cases, where we see resensitivity now to antibiotics, aminoglycosides. With Klebsiella, they have a number of plasmids, get with beta-lactamase genes, but if you can hit it in these large plasmids that have specific genes against antibiotics that the other plasmids don't, I think you have a chance of killing these bacteria. So I do summarize this. I have to do a military conference, so I apologize for this, but if you were looking to kill Klebsiella pneumoniae, I think you would go after these big plasmids, and you would use phages to specifically target them in order to completely eliminate this bug. So thank you all, and I really appreciate getting some time to talk, and I hope I left you some more time for questions for everybody.
Thank you very much, Sabrina. That was fantastic. And why don't we proceed to the list of questions? We do have a few. I do give priority to Gina and Ran. And yes, the clapping is important too. We'll remove the spotlight so we can see others as well. So Gina, you had a question, and I appreciate there's multiple other questions.
Quick question for me, Sabrina. I realize it's a naive question, but how much of the kind of receptor target data do you know about when you are picking your phages for clinical use? Do you consider that when selecting phages, or is all this done retrospectively, the analysis?
Yeah, I think that's more for maybe Maya or Sarah, but I think so far this has been retrospective analysis. But I don't know if Sarah wants to.
Yeah, actually, we are trying to when we have a phagogram with different, for example, phages that are working on our Pseudomonas, for example, we are trying to pick up 2 or 3 phages depending on, yeah, I mean, now we are also doing synergy test between the phages. That's the first point, but we are trying indeed to use phages that are using different receptor and from different families sometimes in order to avoid the phage resistance. Even if finally we are seeing that when we have phage resistance, it might be good, actually.
Okay, good. there's a global question. Does anyone have experience with bacteriophages to decolonize the gastrointestinal tract with carbamase producing Klebsiella pneumonia? Maybe you already get requests for this, Sarah. That could also be part you would want to talk about.
No, not for decolonization, but I know that one of our colleagues in Saint-Luc did his PhD on this matter. It's Jean-Paul, correct? It's Brieux van Nieuwenhuizen from Saint-Luc Hospital who did this, and I think it was for liver transplant children. I mean, children who were just before having their liver transplant, because there's sort of agreement between Brussels and Algeria to treat some patient, and there was this kind of question. But actually, I don't know a lot about it, to be very honest. Jean-Paul, if you have any comments on that.
Yes, so I think they are developing this, but it just did some like a gut on a chip experiment, not more. But they have plans to actually do this one because they're confronted with children with which have a lot of multi-drug resistant bacteria in the gut, and that causes, of course, complications after transplantation. So it's a project they have, but
yeah, just to quickly add, one of our GCPRs was from the Zurich institution at the University of Belgrade, where they're looking at stool transplant plus phages at the same time, and with a special attention to Klebsiella and E. coli. So we do look for some information from them in the future. And what's also great, just to put a shout out to Shauna McKillen, she's the one that has coordinated this global effort for the phagistry and is leading the charge from a scientific side at the University of Belgrade. So it is a great village. So I'm also seeing a question from Sylvia. Sylvia, why don't you go ahead and ask the question?
Sure, thank you so much. It's so impressive to hear about all of this. My question would be about the magister preparation. Would you have to perform all steps in the GMP room, or is it like only the last steps?
So the phage API production does not need to be performed in a clean room. We do because we have the clean rooms for other products, cell therapy products, for instance. So of course we use them because we have them, but monograph does not ask for clean room. It's not mentioned one time. But of course you need to have a controlled environment. But then when this API goes to the pharmacies have their own rules for production of magisteral preparations, which at this time do not require a clean room in Belgium. And then they have their own rules. And they don't have to do this personally. This is something a pharmacist can outsource, but under his or her responsibility.
I see. Thank you so much. And also thank you for all your help with the guideline.
You're welcome.
Okay. We're now in our last minute. Any burning questions that want to come forward? Here's your chance. Okay. If we could just also, please feel free to block your video, show your video so we can give a salute. I believe this has been an exceptional session and will set the pace for future sessions. Also allow future journal forum opportunities. There's over, I think, 50 people that were online at various times today. Great feedback from everyone. So, let's give a round of applause to our speakers today. And as we move forward to the next GCPR, that will depend on the global community because we don't have anyone picked at this time. I think we're wanting to enjoy our summer in North America, our winter in northern, what I'm trying to say, northern hemisphere summer, southern hemisphere winter, and I look forward to our next session. Bye for now and take care.